You guys are what makes this conference possible. So if you guys are going to come in, head in, and if I could have some people close those back doors for me, that'd be awesome. Our first speaker of this last session here is Katharine Seipel. For 2 decades, Katharine Seipel has provided consulting services to a variety of industries, primarily focused on bioremediation strategies and mine geochemistry. At Enviromin, she has applied creative geochemical solutions to successfully support numerous mine permitting projects. With a background in microbial ecology, Katharine continues to be an advocate for incorporating microbial solutions to mitigate the environmental impacts of mining. So her presentation is titled Advances and Opportunities in Microbial Ecology of Mined Environments. So here's Katie. Welcome her with me.
Thanks, Steve. Microphone works. Awesome. And thanks for all of you guys for sticking around today. I know it's getting to be the end of the day, and we're all excited about that social. It's been a lot of tough acts to follow today, good talks, and so thanks to all those prior presenters, including my friend Steve Dent, who primed some of what I have to talk about today. At Enviromin, I think some of you are familiar with the company and the work that we do. We do provide geochemical solutions to mining clients, but we have a lot of passion and also provide services for biogeochemistry in mined environments. So what we're hoping to talk to you, I'm going to talk to you today about, is some advances and opportunities and why we care about microbial ecology of these mined environments, and hopefully how we can help you. Before I get started talking about mining, I want to start talking about microbiology. And I'm wondering just what people think of when they think of microbiology. I think society thinks of infectious disease and antibiotics, hand washing. Maybe you think of a mad scientist in a lab coat with petri dishes, growing all kinds of organisms. And maybe you think of genetically engineered or modified organisms. Maybe you've heard of designer microorganisms that can solve all your problems. And maybe that translates into mining a little bit too, because microbiology in mining has traditionally focused on a few key organisms. Water treatment has branched into a little broader understanding of SRBs that my friend Steve talked about today, and I'll talk a little bit about that too. But key organisms that are focused on mostly in the microbiology and mining field is Thiobacillus feroxidans and Leptospirillum, feroxidans, because we've found them good in terms of bioleaching and bad in terms of when they are helping to accelerate your ARD, or acid rock drainage. But that's not really what Enviromin thinks of when we think of, strictly speaking, microbiology and mining. We have a much broader perspective because there are millions more organisms out there. And so what we key in on at mine sites is the microbial ecology. So broadening that concept and zooming out a little, not just 2 organisms, but all the organisms that are present and how they interact with each other and how they interact with the environment in which they live. That is ecology. So at a mine site, microbial ecology is really key because it means how are those microbes interacting with your rock and your mine-affected water and with each other. And then for our benefit, how can we use that understanding of those systems to optimize the outcomes we want? In the concepts of microbial ecology, it's really important to understand that microbes are ancient, ubiquitous, abundant, and diverse. So they're ancient. I know I'm talking about opportunities and advancements, so it seems a little odd to be looking back 4 billion years here. But the reason this is important to me is because microbes are the oldest organisms on the planet. They've been here way longer than anybody else, and they've been participating in biogeochemical cycles all this time, creating the environment that we live in today. We couldn't be here without them. In addition to being very old, They're everywhere. But what does that mean for your mine site? That means that your mine probably has its own unique microbiome. Your facility maybe has a specific group of organisms living there, and that's going to be different than the group that's living at another mine site. Just like every human here has their own unique human microbiome, this is an important concept to consider in microbial ecology of mined environments, because those microbes interact with their environment to drive geochemical cycles. And this happens with a lot of parameters that we're interested in at mines. Nitrogen, sulfur, and iron being the major ones, but trace elements like mercury, for those of you who heard Steve's talk. On the right here, I'm showing the sulfur cycle that shows the transformation of sulfur forms from oxidated forms like sulfate all the way around into the most reduced form, sulfide gas. And each arrow in this figure represents a metabolic pathway that microbes are capable of facilitating. And that's important because we might want some of these reactions to happen at a mine site and not others. So understanding which microbes are there and what they're capable of may be helpful to us. And that's one of the specialties we have at Enviromin. In addition to being old and everywhere, there's a lot of microbes. So I threw up a few numbers here. Numbers of grains on Earth estimated around 10 to the 18th on Earth. Stars in the universe, about 7 times 10 to the 21st, which are big, mind-boggling numbers. Microbial cells on Earth are estimated to be 10 to the 30th, so 9 orders of magnitude more than the stars in the universe. But at mine sites, that may or may not be the case, because the microbes that are present in various environments, they're not evenly distributed around the Earth, right? They find their environment where they're happy and they thrive there. And we all know that a waste rock dump is a pretty harsh environment. There isn't a lot of water, there's rock, there's probably not a lot of nutrient availability to microbes. So the abundance of microbes in those sorts of environments is probably not as high as in a wetland or something. And this concept of low abundance in mine waste is actually been inhibitory to the mining industry adoption of microbial ecology principles, in part because the techniques to extract the genetic material to understand these systems are tricky. And that's one specialty, again, that we at Enviromin have really worked to optimize so that we can use these really great tools even when there isn't a lot of genetic material, a lot of microbes there to work with. The microbes that are there microbes are diverse everywhere. The microbes at a mine site are also really diverse. Because all those geochemical cycles that I had talked about before play a role in mining, we see a lot of those types of organisms at mine sites. This is 2 figures from a project we've done at Enviromin, so bear with me while I talk you through it. On this graph on the left, we have taxonomic diversity. So that's a fancy way of saying how many different types of bacteria did we see in these samples. In the pink dot on the top here, this was our baseline condition in this bioreactor. We see really high diversity here, over 650 different bacteria, different types of bacteria present. We started our treatment, and we see over time on the x-axis, and depth represented by different colors, but that's not critical here, that the diversity actually goes down as the treatment continued. And it really got low in this end 3 years after the initiation of this bioreactor. So you might wonder why the diversity going down is something that we don't mind. And that's because we're selecting for organisms that can do what we want them to do. So this initial batch may have had a lot of organisms that would otherwise be performing metabolic functions we're not interested in optimizing in our facility. And instead, we are creating conditions that drive the organisms to do the things we want them to do. From that treatment reactor, we have this sample on the right. Well, we have 3 samples on the right. We have a shallow above the treatment zone sample in pink or red, and then the blue and green bars represent the treatment zones at mid and bottom depths. And on this axis here, what you see is all these different oxidation reduction terms here. These are different metabolic processes that the organisms present in the system are capable of accomplishing. And spoiler alert, we were hoping to reduce nitrate in this system, and luckily we see a lot of nitrate reducers. And we don't have chemistry to show you, but we saw nitrate reduction also. Sorry, I maybe didn't explain this. This percent is abundance, so how many of these types of metabolic functions are present. So mostly nitrate reducers. We also saw some selenium reduction with selenium reducers that we were interested in, and a whole host of iron oxidizers, sulfur cyclers, and some different carbon cycling organisms. So you might be wondering, wow, this is so interesting. How come we don't do this all over the place already? And I think it's because there's been a little bit of a bad reputation. We get plenty of projects where someone will try to build a passive bioreactor, and it will work for a few minutes or a few months or a few years if you're lucky, and then it will stop working. And so we get asked a lot of the time, well, if we build this thing, are they going to grow, and is it going to work the way we want it to? And the answer is yes and no, because you have to do it right. So at the risk of sounding like a broken record over all these years, we really feel that we need to move away from this black box design of you can just throw some manure and straw in a hole in the ground and run some water through it and it will work. You have to do the homework at the front end to understand the organisms that are there. The reliance on non-native organisms, be it from a designer bug that someone's got a patent on, or a non-native microorganism population from manure. You know, sometimes they have what you want, sometimes they don't. But they might not survive in that environment, right? This is ecology. It's how they interact with their environment. And we often have to feed them some kind of carbon source. We know that straw and wood chips is great because it sort of offers a walk-away solution in theory, but I would rather have a shot of whiskey than a bowl full of wood chips, and the microbes would too. So we have to start considering more simple carbon compounds, and that sometimes means different styles of design for your facility. And I've already said, but I'll say it again, understanding your native microbial community will really make a big difference on the front end, because then you'll know who's there, you'll know how to help facilitate the outcomes that you're looking for in your facility. So I was going to bore you guys with a lot of academic research out there, because I think there is so much interesting cutting-edge research in this world of microbial ecology, particularly in mined environments. But I was advised that that was too much. So I'm just going to point out this one example of this paper that was published last year, because it sort of illustrates the work that we do at Enviromin, and it allows me to show you how you can take an academic project and bring it to a facility scale design, or influence pilot scale testing that can then be scaled up to facility scale. So Chai et al. Tested multiple carbon sources on a natural microbial community from stream sediments. So this wasn't actually a mined environment, but mined environments can impact natural sediments. So it was kind of a clever way to look at what could those organisms do if they were impacted, and how could we help support removal of sulfate and thereby removal of metals alongside of that. So on this graph on the left, there are sulfate concentrations here and a 30-day experiment here on the x-axis. And to cut to the chase, shrimp shell and crab shell were far more effective at facilitating the SRBs, those sulfate-reducing bacteria, in this native population than the straw compost or sugarcane pulp or the propionate that they tested. And in addition to just putting this material in a bottle and testing the removal of sulfate, these guys did a lot of work on the microbial community analysis. This is a bar chart describing the relative abundance, so how many of these organisms over here that you don't need to be able to read, how many of them were present in each of these samples. These materials in the first 5 rows or columns are baseline materials that they used, so you can see they had pretty low diversity. A few organisms, maybe a lot of a few organisms, in the case of shrimp shell. And then after 30 days of their treatment, you see a lot more colored bands, more diversity in this case, and bigger, thicker bands of these known sulfate reducers in the 2 treatments, the shrimp shell and the crab shell, that worked the best. So why does this matter to us? Because we do the same kind of work. I'm going to give a couple examples of some projects that Enviromin has done to incorporate some of this knowledge. The first one being a sulfate reduction project in a neutral mine drainage. So remember how I said people sometimes come to us and tell us their system isn't working? Well, this is one of them. This was designed as a passive water treatment system for their high sulfate, pretty low metal neutral mine drainage coming out of a waste rock dump. And the water was being collected, as shown in this conceptual model here, and then gravity fed into these buried trenches. So these were pretty large pilot, I would call them pilot scale, trenches, but they were buried underground to maintain suboxic conditions better. And they are designed with an upflow configuration, so the high sulfate water comes in, and the trenches are filled with sheep manure and straw as their substrate. And in theory, the bacteria in that sheep manure will reduce the sulfate to sulfide, and that sulfide will be available to complex with metals and drop out of solution, and you'll have clean water coming out the pipe at the end. But it wasn't working, and it almost never did from the onset. And so we wondered, why isn't it working? Actually, the client wondered, why isn't it working? And we said, well, it could be the community that you have in there actually doesn't include sulfate-reducing bacteria at all. It could be that you're providing an inadequate carbon source. It could be that you don't have enough metals to complex those soluble sulfides, and that they are just coming back out and reoxidizing to sulfate as they come out. Or it could be that the flow is not acting the way you want, and it could be just a flow problem. We did a lot of work with this client to address all of these avenues of inquiry. We were able to show that their sheep manure inoculum did have SRBs in it, and we did a few lab scale tests to show that they're capable of reducing sulfate to sulfide under different carbon sources. So it seemed like maybe it was the carbon source, at least, was part of the issue. But that work led into us running a column experiment with a couple bioreactors to see if we could optimize their system for them. So the gold brownish dots and lines are 2 replicate treatment reactors that we conducted in our lab with their material, their substrate for the inside of these trenches, as well as the black line, solid black line represents the influent water, which was from their site. So replicating the concept of these trenches in a controlled laboratory setting. And on the top here, what you can see is that this is dissolved organic carbon. We saw a little pulse of dissolved organic carbon rinsing off this material when we started these tests. And lo and behold, we saw corresponding sulfate reduction with it. But I don't need to tell you to look a few weeks later to see that all of that just stopped. It only lasted a few weeks, even in our controlled column setting. So we let these run for a while. Could they reach lower ORP and stimulate some sulfide reduction? Would the carbon become available again? What's going to happen if we just run this system for a while and see? And as you can see, for 6 months, nothing happened. Then we decided to add the carbon source we knew from previous testing these organisms were capable of using. And so we added some methanol. And it triggered an immediate response in the sulfate reduction and sulfide production and the reduction in ORP. And in this way, you know, if our client had looked into some of these questions ahead of time, they could have built this system a little better. And now they have the opportunity to modify their treatment system to get the goals that they set out to get from the get-go. But that was a big chemistry project, right? You didn't see any microbiology there, did you? So I'm going to pivot back to talking about genetics. So I think that during COVID, we all learned a lot about like PCR and DNA, and you know, people are starting to be more familiar with these things. So what we do at Enviromin is we are specialized in extracting DNA from low abundance materials at mine sites and running bioreactors, understanding the microbial community through sampling from within the bioreactor of the community that's present. We can do all kinds of extractions of not only DNA, but also RNA and other genetic material that helps us understand not only who's home, but what they're doing and how much of it can we expect to help optimize the goals for our clients. Similar to the sulfur cycle I showed you before, this is a nitrogen cycle. So these are the different phases of nitrogen that you see naturally in the environment. And all of these arrows represent metabolic pathways that microbes are capable of. So we have, you know, nitrogen fixation. I think people are familiar with a lot of these terms, but what we really look for commonly in mine sites is denitrification, to remove nitrate from the system. And that doesn't happen by a straight line here from nitrate to nitrogen gas. It happens as a stepwise process. Microbes can use different sources of energy, oxygen being the most favorable because it provides them the most energy if they're capable of using that. But a lot of oxygen, or a lot of organisms in the environment, can also use other sources of energy. And in this case, we were interested in some facultative anaerobes that are common in modern environments. They're able to switch from using oxygen to nitrate. So if you just remove some oxygen from your system, they'll switch over to using nitrate. And now I get to show you the output of a genetic, some of the genetic analyses we do. Unlike the colorful bar chart that I showed you before that was saying how many organisms are there and in relative abundance, what we're looking at here is what genes are present. So those metabolic pathways, the arrows that link the different types of nitrogen species, are facilitated by enzymes. And those enzymes are coded for by DNA. And these are genes that represent coding for these different enzymes that complete these different steps in that denitrification pathway. The different sizes of the circles here are kind of what matters in this graph. The bigger ones say that there's more of those genes present. The smaller ones mean that there are less. And what I want you to note here is that for the step from nitrate to nitrite, we have a lot of big circles. And nitrite to nitric oxide, we have some, medium, pretty good size. From nitric oxide, to nitrous oxide, we see big green circles. And then this final step, converting this greenhouse gas, nitrous oxide, to an otherwise innocuous gas, nitrogen gas, it's actually smaller circles. So with this information, we're able to say, well, maybe we need to tweak the system and figure out if we can optimize the final steps to make sure that that nitrate is removed as nitrogen gas. And so that's just a glimpse into the world at Enviromin. And I bet you're now wondering, how do we grow this application? How do we use it more? How does this relate to what I do in mining? And the answer is we can incorporate it into lots of phases of the mine life cycle. So right now, there's a lot of focus on closure and remediation, water treatment, maybe abandoned mine lands, or you're trying to close a site, so maybe you want to think about how to get your water clean and not have to pay for active treatment into perpetuity. And the answer really lies with, investing strategically and incorporating some of this novel technology as well as others. This isn't limited to microbial ecology, into permitting and the construction and operation. I think my friend Matt keyed in on this. We have to learn ways to do things better and different. And it might not be always easy or cheap. It might not always be the simplest solution, but if we can start building smarter mines that build water treatment into waste rock dumps that happens during operation, then you have better opportunities for walkaway closure scenarios. And I will just take a moment to say that my colleague, Dr. Kirk, recently just finished a publication that's accepted into the Science of the Total Environment. We'll have a link to that on our website shortly. With that, I'll take questions.
All right, questions for Katie?
I know you talked a little bit about designer microorganisms, but I'm wondering if there's any work that's being done to introduce desired genes or metabolic pathways into the natural community by plasmids or other vectors?
That's a great question. I love that word, plasmid. See, she knows what she's talking about. We don't do that work. There could be academic research. I wouldn't be surprised if there are academic research facilities looking at saying, we know we want this type of genetic, these metabolic processes in these organisms, and we know they can live in this environment, so let's insert them in there. That's what a plasmid is used for. So the answer is we don't do that. But yes, that you're exactly keying in on this sort of divide, which is not unique to microbial ecology, between academic research and applied science. So it's a hard sell to get from one to the other often.
Thanks, and great presentation. I had just a question about your carbon source. Some of these mines are very, deep in the mountains, very remote, hard to get to sometimes. Mm-hmm. And, methanol's a great labile carbon source. Are there, rather than just wood chips or
right.
or straw that are kind of more complex and maybe not as accessible, is there other alternatives that could be more of a at least partial walk-away solution?
Well, I was really excited about those crab shells until you told me about the mercury today. The answer is yes and no. I think sometimes we may be looking to solutions where you might be able to do a pulse, a semi-passive system where you have to maybe re-stimulate the system occasionally. I am not aware of any more, slow release mechanisms that are available at this time. But yeah, I think that's a big hurdle for these kinds of applications in remote sites.
I had a question about temperature changes and the viability of bioreactors, because I have heard bioreactors kind of like dying off in the winter, or if temperatures get too cold or too hot. So I didn't know if that was
I think
something
that's
to
a
come across.
really good question too. I think it comes down to testing as well. If you're able to test that variable in your pilot study or bench lab scale study, you might be able to design your facility a little better to protect it from environmental fluctuations, like temperature fluctuations like that. Larger facilities would be a little more robust. Making sure you have a diverse community in your system will allow it to be more robust in the face of changes. And there are other types of upset too, like a freshet, like a pulse of cold spring water into a system could really disrupt the system as well. But temperature flux is a good question as well.
All right, any more questions? All right, join me in thanking Katie.
Thanks,
Great job.
Steve. Thanks, everybody.